Okay, hello everyone. Welcome to today's webinar, which is the first of a series of webinars organised by IPEM's Special Interest Group for Clinical Scientific Computing. It's brilliant to see so many people here. I can see there's still more coming in, but we'll get going so that we kind of stick to time today. We've got two really great speakers lined up, thinking all around maximizing the use of our data. My name is Josh Kirby. I'm one of the members of the kind of special interest group for CSC with IPEM, and then James Mogridge will also be helping out with the questions and things today as well. Be one of the faces you can see on the screen. Just a couple of housekeeping things before we start. The webinar will be recorded, so if you don't want kind of your camera to be recorded, etc., I would recommend keeping that off, and it will be available on the IPEM Conferences YouTube channel at after the webinar, so you'll be able to kind of look back and see of anything that you missed. What we are going to do today to try and gather questions in kind of the best ways that you can kind of upvote ones that are similar to yours, etc., is we're going to be using Slido for Q&A. So you should have hopefully got a link as you joined, but if not, James from IPEM will put that in the chat so that you can access the Slido to be able to add questions. And feel free to add questions on the Slido as you go through, and then myself and James will kind of go through those and try and get as many of those answered after the speakers have kind of each speaker has spoken. And if we don't get through any of them by the end, we'll try and get that information for you. I guess really, rather than listening to me, let's go straight into hearing from our speakers. So our first speaker is from the UK Biobank, and it's a senior data analyst, Laura Boteva, who's going to talk to us a little bit about using data on quite a massive scale. So over to you, Laura.
Thank you, Josh. I'll just share my screen if that's okay. Oh, sorry, that's right on screen. Sorry about that. Oh. Okay, we just tested it and jinxed it, didn't we? Sorry, maybe I'll just stop sharing for a second and then. Can you see my screen at the moment or no?
Not yet. No, we can just see a blank screen.
Okay, just give me a second and I'll test it in a second. Sorry. We just tested it a moment ago.
Yeah.
We thought we had it covered. Let me try it again. How about now?
Yep, we can see that now.
Perfect. Okay, sorry about that. So I would just like to start by thanking to the organizers for giving me an opportunity to talk a little bit about our resource. And I also just want to mention that a colleague of mine, Dr. Joe Gay, is also on the call. The reason being that the UK Biobank data analyst team is split between sort of the Manchester side where I work, and also some of the team is incorporated in the Oxford University as well. So we cover slightly different aspects of the resource. So hopefully between myself and Joe, we should be able to answer most of your questions. So just in terms of, I'm sure that probably many of you will be familiar with UK Biobank, but just for those of you that may not be, it's a large-scale biomedical database. and it contains quite in-depth genetic and health information from 500,000 UK participants. So we augment the data resource regularly with additional data, and we also make it accessible globally to approved researchers, the condition being that they need to be undertaking health-related research. So we like using this slide just to give people a sense of the scale that we're talking about. So this is our automated sample store located in Cheadle, Manchester, and it holds about 10 million samples, which is about half the number of samples we have collected from participants since the study started in 2012. In terms of what the study includes, it was, so for each participant, it was established with what we call the baseline visit. And that was conducted in one of our participant centers across the UK. What that visit covers is, first of all, a touchscreen questionnaire, where participants fill out a questionnaire focusing on things like their lifestyle, various socio-demographic factors, family history, and more recently we have also incorporated things like cognitive tests as a part of that. The next they undergo a verbal medical interview with a trained practitioner, and that covers their medical history. We also take physical measures, so things like BMI, blood pressure, ECG at rest and during exercise. Then very importantly, we move on to sample collection. So we tend to collect blood, urine and saliva from participants that are willing and able to donate them. And we have done that for all 500,000 participants in the study. In addition to that, we also have repeated this assessment for 20,000 of our participants. And beyond that, we also have imaging data for about 80,000 now of our participants, and essentially this is acquired through an additional visit which we call the imaging visit, and that covers MRI imaging, a repeat of everything that happened at the baseline visit, so things like questionnaires, medical history interview and sample collection as well. So for about 8,000 of our participants as well, we have performed repeat imaging. So they have come for 2 imaging visits, and we are hoping that by scaling this up to 60,000, which is the plan, we'll be able to create the world's largest longitudinal imaging data set. As part of that as well, we've got the so-called COVID repeat imaging dataset. So this is focused on participants that had an imaging visit prior to the COVID-19 pandemic, which then were asked to return for an additional imaging visit, and that covers about 2,000 of our participants. So obviously, the longitudinal aspect of the dataset is something that is quite important for us, and you can see on these plots the total number of visits we have for participants. So for about 100,000, close to 100,000 participants, we have more than one visit. And on the left-hand side, sorry, on the right-hand side, you can see the time between visits and sample collection. And as you can see, we have quite a range of variation. So we've got some participants where we've got about 15 years between 2 subsequent visits, and for some participants, it's as little as 2 years. So it really gives us quite a lot of longitudinal information. In terms of how we regularly augment our resource, there is a variety of modes where we do that. So one way is through web-based questionnaires. So participants fill out questionnaires online through their phones, and these tend to be focused on things like lifestyle factors, mental health. We have dietary questionnaire. There is a whole range of questionnaires that participants fill out, and we do send them regularly as well. So for a lot of participants, we have more than one instance of the questionnaires. We also have a couple of studies that incorporate wearable devices. So we have a data set for focused on accelerometry, where 100,000 participants wore accelerometers for a week. We also have repeats for about 3,000 of those. We have 4 additional instances where they repeated that process across different seasons to account for changes in seasonality, and again, enhancing the longitudinal aspect of the data set. We also have an additional wearable project ongoing at the moment, where we've asked participants to wear cardiac monitors on their chests for 7 days, and we are accumulating the data for that at the moment. Another major source of data coming into the dataset is through ongoing NHS linkage. And essentially that covers things like primary care records. We have that for about 40% of the cohort. We also have data on things like prescriptions and hospital admissions. So all this data tends to come up using a variety of medical codes. And what we aim to make, we make it available with the original medical codes, but also one of the biggest aims is to make it accessible to researchers that may not necessarily have background in understanding medical coding. So we also provide a data set using OMAP coding, and also we provide a very easy to use sort of algorithmically derived data set that includes the data in a very easy to use format. For example, things like when is the first date when a particular participant was diagnosed with a certain ICD-10 code. Another major source of additional data for the dataset is, of course, our samples. And the genomics dataset is one of probably the most well-known and the most widely used by researchers. It has been going on for a while, and every release we do sort of reflects the current technologies at the time. So initially, the genetic data that we had for participants was limited to genotyping data for the full cohort, and that is essentially a targeted method that only interrogates genetic variation at regions of where we know genetic variation exists. We expanded that with sequencing data that was focused on the protein coding parts of the region, of the genome, sorry. And most recently, we released full genome sequencing data for the full cohort, so 500,000 people, which at the moment makes us the largest collection of genome sequencing data in the world. Beyond sequencing, we also have additional ongoing sample projects. A dataset that has been very popular with researchers is the proteomics dataset. So this is again a targeted method that looks at the levels of 3,000 proteins in serum, and that was performed for about 60,000 participants. Going back to the longitudinal aspects of the dataset, samples from the COVID repeat imaging visit that I mentioned earlier will be included. And just as an example of how we tend to select samples for these large-scale projects, we have a few different subsets in terms of sample selection. So we have a subset of about 46,000 participants, where we try to essentially make sure that that subset reflects the participant characteristics at the baseline visits. We have an additional set of about 6,000 participants where they were selected for particular conditions or for having imaging data. And then finally, we've got the longitudinal COVID-19 imaging samples as well. So in our view, the fact that we've got longitudinal aspects is one of the most interesting aspects of our study, and we are always looking for ways to leverage that. And I have a couple of examples of these. So for example, we currently have an ongoing metabolomic study, and that's carried out with Nightingale Health. They're analyzing about 450 biomarkers in plasma, and about 15,000 participants will analyze repeat samples from them. So I have a plot here of the average gap in years between the two sample collection dates, and just underscoring how prospective studies can work in that way. Within the time frame between the two sampling dates, there were about 25,000 new diagnoses across about 8,000 participants, with 600 distinct ICD-10 codes. Most of the new diagnoses were focused on things like hypertension and metabolic disorders as well. But it really means that if researchers want to use that data set to focus on particular diagnoses that's included in those 25,000, then they have that longitudinal data prior to and following diagnosis. And this is obviously not something that's happened by design. So another interesting data set that leverages the longitudinal aspect of the data is the neurobiomarkers data set, which was released in November. So this data set looks at concentrations of 4 neurological degeneration markers measured in plasma samples that were acquired as a part of the COVID-19 repeat imaging visit. So it allows to look again at the concentration of these markers in participants prior to and following COVID-19 infection. So we have that data set for about 1,200 participants, and a lot more. All of these participants have MRI data prior to and following COVID-19 diagnosis. And I have an example here on the left of how that data was leveraged previously. So there is an example of a study which looked at the changes in brain associated with COVID-19 infection. But obviously, by adding this additional data set, it means that we can then add a molecular dimension to that data set as well. So generally, what we have here at UK Biobank is a complex data set that is continuously evolving, and that's both in terms of the quantity of data, but also longitudinally. So it's types of data and the longitudinal aspect as well. Another interesting but very challenging thing is that it enables the use of diverse data sets together. And this really means that there are types of research that can be done with our data that can't be done with other data sets. So one example is, I have a couple of examples here in terms of public publications, but one of the high profile papers is looking at the genetic influence on brain phenotypes. And obviously, because we have the genetic data and the medical imaging data, we are in quite unique position to provide researchers with data to do this type of analysis. The one at the bottom uses the proteomics data set that I mentioned earlier. And again, that looks at association between protein levels and genetic variation as well. So again, the type of data set, the type of studies that can only be undertaken with really comprehensive data sets like ourselves. So obviously, having such diverse data does provide quite a lot of challenges. In terms of usage, we've got about more than 30,000 registered researchers across both the UK, but also the rest of the world. And obviously, with such a large sample of researchers, this spans researchers at multiple levels of their career, multiple fields of expertise, and also multiple levels of resources in terms of computational resources and things like that. So in terms of size and complexity, I'm sure you can imagine how complex the data set is. So I have a plot here of the volume of data that we are holding. So the WGS at the top is the whole genome sequencing data set. And as you can see, it absolutely dwarfs things like the imaging data set, which is very, very big in its own right. So one of the things that makes the dataset even bigger is that we try to ensure that data is available reflecting every stage of the analysis process. So I don't know if there are any bioinformaticians in the audience, but I've got an example of the genomics data here, where we have the file types generated throughout the full pipeline of genomic analysis. So we have CRAN files, which are sort of the raw files where the genetic sequences are aligned to the human genome. We have VCF files that are focused on the variation. We have PLINK and BGEN files, which again are focused on the variation, but they're designed to be used with particular pieces of software. And of course, we always try to make sure that this data is available as well to researchers that may not have bioinformatics background, but might be interested in incorporating some of that data within their analysis. And we tend to do that through sort of tools developed in-house that we publish on public repositories, like, for example, GitHub. And this is one way that we try to address the fact that we have user base that is quite diverse in terms of skill sets and fields of expertise. So one big challenge with our data set as well is that obviously it contains a lot of personal data for our participants, and it's very important for us to safeguard that and to make sure that it's used responsibly. So we have two models through which researchers access data. So for some of our data set, we allow researchers to download it onto their own devices. But for the genetic data set, we only allow that to be accessed in our cloud analysis platform, which means that it means that we can balance accessibility, usability, and safety. One big challenge that we're struggling with there a little bit is things like enabling reproducibility and cross-cohort analysis. So obviously people might want to integrate our dataset with other datasets, which might be enclosed in their own research environments and might not be available for downloads as well. So there hasn't been that much research looking at how best to address that. But what I'm showing you here is a study from 2022 where people looked at sort of the role of genetic variation in, I think, lipid metabolism, and they compared two ways of analyzing the data. So one way was via meta-analysis, where they combined the dataset from UK Biobank and the American study All of Us, and then they looked at which regions of the genome mostly contributed to various phenotypes involved in lipid metabolism. On the other side, on the right-hand side, is a pooled analysis that essentially looked at the summary level statistics data and went from there. So that's the type of analysis that does not require the raw data for the raw data sets to be pulled together. And actually, the results are very, very comparable, which would indicate that it is possible to overcome the challenges associated with accessing data that's within sort of a protected research cloud-based environment. Just to focus a little bit more on our cloud-based platform, it's a cloud-based platform that's hosted by DNA Nexus. And essentially it enables users to create projects, share code, and share analysis tools. They can perform downstream analysis and just create pipelines that can be reproducible across projects. We also, in terms of like the way to access and analyze the data, we provide quite a few different methods to do that. So they're more graphical-based methods for researchers that may not have like data analytics or bioinformatics computational background. We also provide command line interfaces, cloud instances, and things like that. In terms of sort of the command line interface, it's very similar to how sort of HPC clusters work. So it means that a lot of the code that researchers might have developed for like HPC-like environments will translate quite well onto the wrap. So in terms of one of the reasons that we like having the platform as well is that it really enables us to democratize access. So I mentioned the size of the genomics data set, but essentially it would mean that most researchers would not have the computational capacity and the storage capacity to be able to download and analyze this data set in-house. So essentially, it really means that we are enabling researchers to use that data set that may not have the capacity to do that otherwise. It does have a lot of other advantages as well. It means that we can update data regularly and provide that to researchers without needing the researchers to download the data. It's very flexible, so researchers can request various instance types with various computational capacities. So it really means that it's very scalable. Yeah, it also provides, as I mentioned, it does provide a variety of interfaces. So yeah, ultimately it does democratize access and hopefully lowers the cost of research. And yeah, so I think that's it. I'm happy to take any questions. As always, I would like to just thank everyone else in the UK Biobank data analyst team and of course our participants.
Thank you very much, Laura. We have some questions that have come in via Slido, so if you don't mind, I'll ask the first one.
Sure.
How is your data sanity checked? E.g., if you have people with blood pressure recorded at some level like 500 or they're age 300 years old, do you have ways in which you can sort of detect that sort of submission before it enters your data set?
So I think we are definitely in the process of sort of optimizing our pipelines. So at the moment, we are mostly doing sort of ad hoc checks on the data where we look, so the data is sort of automatically uploaded to our database, and then we might sort of check it afterwards, do sanity checks. We don't have sort of automated filtering or anything like that, at least not for the data that comes straight from the participant visiting centers. I think we are in the process of building those tools, allowing us to kind of monitor the data coming from the centers on the day-to-day basis, but we're not quite there. In terms of the sample projects, we are definitely a lot further ahead in that, just because the data for the way the data is returned for the sample project is a lot less challenging. So the data for those tends to get returned a couple of times a month or once a month, and it gives us the chance to really sanity check it before releasing it publicly. And for sample projects, we normally have things like specified QC criteria that are agreed with the researchers performing the analysis ahead of the project starting, and we monitor them kind of on an ongoing basis to assure that the methods do live up to those criteria. And of course, if they don't, then we don't upload the data for public use. I would say as well, on the other hand, we try not to be too stringent as well, because we do want researchers to give researchers a chance to perform their own QC, to perform their own filtering, just because of course they might be able to discover something there that they might not be able to discover if we've pre-filtered the data.
Thank you. Just as a quick follow-up to that, do you apply similar techniques to your imaging data inputs as well? If you're trying to standardize across sites and longitudinally?
Yeah, of course. So definitely for the imaging data, we monitor that on a daily basis, and we look at various sort of indicators. Yeah, so I'm probably not the best person to talk in depth about the imaging, but yeah, we absolutely have ongoing checks for the imaging data, and we monitor what comes out of the centers every day, and we definitely look at outliers and follow them up with radiographers at the imaging center. It is one aspect where we actually do quite a lot of checks, just because obviously if a scanner is not performing well, it is a huge drain in terms of cost, participant time, staff time. So it's absolutely something we take very, very seriously. Yeah.
Thank you. There was a few questions regarding consent. I see that I think Joe has replied to them in the chat. But I don't know whether Joe or yourself, Laura, want to add anything further to that regarding opt-out, and also when you have events where data is changed, whether there's any option there, how participants are informed and can consent to that, if that makes sense.
So opt-out is sort of participants withdrawing from the study?
Yes, yeah.
Yeah, so we do have methods for handling that. So as soon as participants get in touch with sort of our participant contact team, that data gets through to the database, and from there we make sure that all of the data associated with these participants is withdrawn from the researcher data. And I think this is one reason as well that we really like the cloud analysis platform, because it does give us that control. I think otherwise we tend to reach out to researchers and give them a list of withdrawn participants and ask them to delete that data from their data set. But obviously that puts the onus on researchers to do that. While with the cloud analysis platform, it means that we can automatically withdraw it from use for all researchers using it. I think the other aspect of that as well is the sample aspect. So when we get participant withdrawals as well, that definitely filters through to the lab, through our team, and the lab makes sure that all the samples we have associated with these participants are destroyed as well within, I think, 2 weeks of the withdrawal.
Thank you. I hope that's answered all the current questions within Slido. I don't see any others that have appeared, but I think we can keep the sort of chat going, and maybe we can touch base again at the end, and perhaps we can answer questions and sort of feedback to the people asking them in our own time. I think at this point it's good to switch over to Will Yule, who's joining us from Hertfordshire County Council. So a different slant on the on data sets and how we manage our data. So without further ado, I'll bring on Will, if that's okay.
Hi, I will just try and share my screen. Hopefully that's coming across
Yes,
now.
I can see
Awesome.
it. I hope
Great.
everyone else can as well.
Cool. As you say, yes, hopefully a bit of a good corollary to what Laura was saying around around data challenges and being more around the variety of data rather than the volume of data. So I'm going to talk a bit about tidy data and reproducible analytical pipelines, which is another RAP acronym. I can't remember what yours stood for, Laura, but hopefully it won't be too confusing for people. And how this is relevant to my role. So in public health, we work with a huge variety of different data sets, everything from kind of healthcare to employment and education and environment and all sorts. And that data is published by different people. Some of it we control, most of it we don't. And that means that it comes in a very, very wide and creatively formatted way, which involves us doing a lot of work to tidy that data and to build ways that we can reproduce our analytics. So that's kind of what I'll talk about today. So just a brief overview of the kind of analytical workflow, which probably people can hopefully recognize. Start off by importing our data and then trying to tidy it, so to get it into a format that we can actually do our analysis on, and that can take a very long time, or it can take a very short time. Hopefully it takes a very short time, often not though. And then we do the process that we kind of think of as analysis, which is to derive some understanding from the data by applying various different methods to transform the data. Maybe we want to standardize it in some way, to visualize the data through plots or maps or however we want to visualize. Potentially we'll do some modeling like regression modeling or something like that. But at the end of the day, we want to be able to communicate the understanding that we've gained from doing that analysis. But what we're often met with when we're doing that importing and tidying step is something like this. And this is just an Excel file that I took a screenshot of from online. But if you're anything like me, you'll see that and be a little bit, where do you start? There's a huge number of colors. There's lots of empty cells. It's very difficult to really understand what's going on. As well as the empty cells, there seems to be a few like NAs, and there's these merged cells and some numbers at the top that presumably have some meaning, but it's very, very difficult to really understand what's going on just to get to the point where you can do any analysis to derive any understanding from data like this. And I'm sure that people are familiar with data sets that have been sent to them like this, and possibly even created for you themselves. And that kind of chimes with how data scientists say they spend their time. So there was a survey done a few years ago that found that 60% of a data scientist's time was spent doing data cleaning and organizing, and rather than actually getting the understanding from data, which is where their skills come in most useful. And part of the problem with that is that while tidy data sets are kind of all alike, messy data sets are always messy in their own way, which is essentially the Anna Karenina principle, where all happy families are alike, whereas each unhappy family is unhappy in its own way. And what that basically means is that when we have tidy data, we can use standardized ways to process that data. So we can, have some process that reads the data and then analyzes it and models it and visualizes it and communicates it. But when we've got messy data, we need to kind of bend our tools to fit the data, and maybe make new tools, and maybe those tools are only useful for that specific piece of data and for nothing else, so which can be quite frustrating and also incredibly time consuming and inefficient. So you should probably talk a bit about what we mean by tidy data. There's a guy called Hadley Wickham who wrote an article called Tidy Data, which the name suggests was about tidy data, and he's also the author of a collection of R packages, R being a programming language, called Tidyverse, and a lot of the collection of packages is all about kind of working with and tidying data. And he proposed that tidy data has 3 core principles. The first one, that each variable forms a column. Second one, that each observation forms a row. And the third one, that each cell is a single measurement. And they sound incredibly simple and straightforward, and kind of baffling that any data would deviate from such basic principles. But on the other hand, huge amount, possibly even the majority of data that I work with doesn't. And I'm sure there's lots of data that other people work with that doesn't as well. So I thought we'd get on to an example of kind of working with tidy data, but first of all, I'd go through some of the more common data formatting problems, which are termed the 7 deadly sins, because there's 7 of them. And the first one is where a variable is used as a column header, and it's often done for things like dates and times and things. So we can see on the right here, we've got a table that has a country variable, and then a variable called 1999 and a variable called 2000. You can imagine it having many more variables, 2024, all the way up to there. And although that's potentially quite nice to look at, it's actually quite hard to analyze. If I said that we wanted to find out how many things had happened between 2010 and 2020, you'd have to kind of go in and manually select those columns somehow and sum them up. The other problem is that we're going to run out of columns at some point. Certainly if you're using Excel, I can't remember how many columns Excel has. You might not run out if you're doing it in years, but if you're doing it in hours, you're probably going to run out relatively quickly. And the third issue with this is that you don't really know what those values represent, because there isn't a column, there isn't a name for what those variables are. And you can see if we pivot it into a longer format, where we create a variable called year, we can give the numbers a name, in this case, cases. Second common problem is that one observation is scattered across multiple rows. So we can see here that we've kind of got a variable called key, which has got cases and populations. If we wanted to create a case rate, for instance, it would be quite difficult, because we'd have to divide the first value of the value column by the second value of the value column, and the third value of the value column by the fourth value of the value column. Which is going to get a little bit messy. There's probably quite a lot of potential for error to creep in. If you're thinking about Excel, someone goes in and inserts a cell, and it changes all of your cell references, and suddenly you're doing the rate of cases in Afghanistan in 1999 by the rate of cases in Afghanistan in the year 2000, which is not really what you would want. So what we can do is, instead of having one variable that isn't really a variable called key, we can create 2 proper variables, one called cases and one called population, and make our data wider in effect. Third common problem is that more than one value is stored in a cell. It's a little bit of a contrived example, but maybe I think especially when you're working with tables of data that are meant for communication, you can often find this. And also with categorical data. There's some data I was working with, I think it was last week, where I wanted to be able to find everyone who was discharged. But there wasn't a discharge column, there was a kind of an outcome column. And it was essentially formatted as on the slide here, where we had discharged care home, discharged home, and there were lots of other options. If I wanted to find everyone who was discharged, I'd have to go through and select all of those options. And if the data changed and a new option was created, I wouldn't necessarily be able to have that in my data. So I'd have to continuously check to make sure that I had all of the discharges selected. Next one is around one variable being stored in multiple columns. Again, a little bit of a contrived example with century and year, but you can probably see it more often with things like day, month, year, for instance. But not too common. Fifth one is around missing data not being coded or not uniformly coded. And we sort of saw this with that initial Excel sheet where we had lots of empty cells, some with NA. I think there were a couple with missing written in capital letters. And we don't necessarily know if the blank cells mean the same thing as the NA cells as the missing cells, although they mean something different. Penultimate one, different data types stored in one column or one variable. This can often be a problem with missing data, in fact. So if we've got a numeric variable, blood pressure measurement, say, and you go in and, if there's a missing value, you type missing, the actual text missing in it, it's no longer a numeric variable, it's a text variable to many programming languages and also potentially to Excel as well. And that can create problems. And finally, and this is the really big one with Excel, is that styling denotes meaning. So we saw in that initial Excel, there was lots of color, there was lots of merged cells, there was borders, there was all sorts of stuff going on to presumably make it easier to understand. I don't necessarily know if that was a great example of something that was easy to understand. But that's very difficult to analyze. How do you extract that this cell is yellow using Excel, or indeed using much else other than your eyeball? And that kind of gets to the root of some of the causes of why data is maybe not tidily formatted, which is that people use tables both to communicate data to end users and people, but they also use tables to store data for analysis. And it can be very, very difficult to go from a table that's been designed for communication to one that's been designed for analysis. So I thought it'd be worth giving an example of how we could tidy a data set. So if you imagine we've been given the task of calculating a case rate per 10,000 people by country for all years combined, and our data looks like this. So we've got a country column, we've got a type column, and we've got these 2 years. And you probably see already that there's a few issues with how that data is stored if we're adhering to tidy data principles. Obviously, we could use Excel to do this. Maybe be a little bit messy. Maybe the most kind of intuitive approach would be to kind of drag and drop some of these cells. It would take you a very long time, but it would get it done. There's maybe some other fancy bits of Excel that are more recent that could help with some of this, but it's probably not going to be massively easy to do, because ultimately what we want to be able to do is to sum the 745 and the 37,000 and then divide them by this, whatever it is, 19 million. We want to sum the 745 and the 266 and then divide them by the sum of these. And instead, what I'm going to go through is how we could use a programming language like R to do this. So R is what Hadley Wickham, the guy who wrote the Tidy Data article, mostly uses, and is the programming language that he's written a load of packages to tidy data in. So to start off with, we're going to load in a couple of the packages that he's written, one called dplyr and one called tidyr. The tidyr one kind of gives a hint of what we're going to do. And we can see the data on the right here. So this is the original unmodified data frame. What we can do at first is pivot it longer. So what we've done here is taken these 2 year columns, and instead of having them as columns, we've made one column. We've made one column that is year and another column that is value. And we can already see it's a little bit tidier already by pivoting it longer. We can then pivot it wider. So what we've done there is take this type column and make it into 2 variables, cases and population. And finally, we can group by country and summarize the data, and we get a rate of cases by country across all of the years of the data. And hopefully you can kind of see how you could use that code to do the exact same process on an updated data frame in the same format, or even data in a similar format, and potentially be able to reuse some of these functions like pivot longer or pivot wider on completely different data sets that are formatted in very different ways. So yeah, so obviously used a programming language to that. I don't know how familiar people on the call are with programming languages. I thought it would be worth doing a very quick bit on why you might use them. So one of the big things about them is they're reproducible, so we can rerun and reuse code, as we've kind of seen. They're very expressive, so we can create new concepts. So you could think about trying to tidy that data in base Excel. It'd be quite difficult because the people who built Excel haven't built a tool for you to do that. In programming languages, we can reshape how tools work. It's diffable, so we can really quickly find and track changes, and that is massively underappreciated or overlooked benefit of programming languages. You know, in Excel, once we've done something, it's done. You can maybe undo it with Control Z or something, but if you saved your file, you can't really go back and see what the process has been to take that raw data and create your hopefully tidy data. Programming languages, we've got the text, so we can go back and really understand what's happened. Related to that, it's very readable. So, you know, even if you don't know R, you can probably get somewhat of an idea of what's going on with the processing here just by the naming of some of the functions that we've used, like pivot longer and pivot wider and group by and summarize. And it's also very shareable because it's text. We can just share the text, and we're not hopefully going to breach too many privacy rules. And what that means is that we can save time by reusing our code and also reusing the code of others through functions and packages like tidyverse and tidyr and dply. And we can also transform that analytical workflow into a reproducible analytical pipeline. So the different thing that RAP stands for, other than RAW thing, is reproducible analytical pipeline, which is essentially a framework that tries to take that analytical workflow that we talked about at the start and make it as repeatable and automatable as possible. So in the non-RAP approach, we might have our database and then we use SQL Server to pull the data into Excel, and then we load the data from Excel into SPSS and do some analysis of some form, and then we export the data into Excel to make a slightly prettier graph than the ones that SPSS give you. And then we copy and paste that graph into Word, and then we PDF the Word document, and then we email it out. What we can do with programming languages like R and Python is just use packages that people have written like Quarto to do that entire process in one file or a set of files and one process. So these slides are written in Quarto, and we do a huge amount of our analysis now in Quarto because it means we can have one file that both pulls the data, analyzes the data, or pulls the data, cleans the data, analyzes the data, and communicates the results without us having to like copy and paste and move things around manually. And that allows us to be really repeatable and really automated about how we do things. Put a couple of links at the bottom of that slide about doing data science and reproducible analytical pipelines. So one is about a radiotherapy data set that was talked about in a community called NHSR a few years ago. They had a conference, I think it was the 2022 conference, that I thought might be relevant to this group, although there's lots of other talks that they've given that could probably be related as well. So that's essentially it, and hopefully gives you an idea of how you can start to build pipelines that ingest data, wrangle it, visualize it, model it, and communicate that completed analysis to other people. There's put in some slides around resources and tools. So if you're an R user, the tidyverse is the kind of the big thing when it comes to tidying and working with tidy data. If you're more on the Python slant, if you do use programming languages, I'm less familiar, but I believe Polars is getting to be almost as good as tidyverse in many ways. And there's various other resources there about reproducible analytical pipelines. And finally, some acknowledgements to the various images that I've stolen and code that I've adapted as part of this. But I will leave it there and hand back to you, James or Josh.
Thanks very much, Will. It's really great to hear kind of the two sides really. We've got the kind of the massive kind of data projects from Laura, and then the kind of more day-to-day kind of practicalities that you've talked about there, which is really good. Someone's asked about whether we can get links to the resources on the slides. I think we can definitely get those kind of shared or sent out afterwards. I think as well. I did have a question around, so I guess you were talking about how lots of the data you use is not data that you control or have kind of control over how it's recorded, and so as a result you get it in loads of different forms, etc., which I suppose is not something in your position you can change, and so most of it is on how you then deal with it. Do you think there's a need for kind of improved education from people to change people's views on what a database is for? So in terms of, because you said about the two different things, you've got the human readable and then the what's useful for data analytics. And presumably we need to educate those who aren't doing the data analytics but providing the data so that it's then in a form that's kind of easier to use. Is that fair, or would you say something different?
Yeah, absolutely. So there's kind of there's some tendencies to move towards more open data, but often those are publishing things in a CSV rather than an Excel file, which doesn't necessarily provide a huge amount of benefit. And there is some movements to doing things through APIs or application programming interfaces, which can be much better depending on how they're implemented. But as you say, the big thing is getting people to publish data that they want to communicate in a way that's really good to communicate, and publishing data that they want to be open and available for analytics in a way that's good for that, and those being quite different things. So yeah.
If anyone has any more questions, do pop them on Slido. Another question would be, obviously you mentioned about missing data, and obviously that's a big challenge. When, particularly in public health, I guess you're, again, separated slightly from the clinical context. How would you go about making a decision about what to do about missing data, in terms of, do you kind of leave it blank, put some kind of a standard default in? How do you deal with that within the kind of that separated context?
I think it really depends on the project. We do try and work more with clinicians and healthcare associated people now than we did before, rather than being kind of the public health ivory tower. So it really does depend on the project. A lot of the time, because we're working at a population level, we're more interested in trends and things that apply to not necessarily individuals, but in general. So in a lot of cases, we don't necessarily lose a huge amount by dropping it. What we can also do is, because we've got access to so many different data sets, is see if we can link stuff in. So the NHS recording of ethnicity can be patchy. It can also be patchy in the local government recording, but they're not necessarily the same people who are patchy. So we can do some kind of combinations there, although then you usually end up with the same person being Pakistani in one data set and white British in a different data set, and how you deal with that can be a bit of a challenge.
We have had a question come in on the chat actually, which is how are the tidy rules different to COD's relational database rules?
So they're probably highly related, and there is it's not necessarily in Hadley Wickham's tidy data principles themselves, but he does talk about storing related data in one database and not storing everything all in one database, and when to separate tables and things like that. So I think they're highly related, but maybe more specific to an individual table rather than a data schema as a whole. I mean, if you're doing things like that, you're probably more on the storing data properly than many people, I would imagine. But that's not to say that, you know, especially around having the correct variables, that, you know, is the kind of thing that can still catch you out, even if you are using good database principles. So I think they complement each other quite well.
That's really great. I don't see any more questions. Well, there are a couple of extra questions for Laura though, so we might pop back to Laura, and then as we've got a couple of minutes. So there was one that said, how representative is the full population is kind of your sample within the biobank? And does the fact that it's opt-in mean that there's kind of it's susceptible to self-selection bias?
Yes, so it is a very frequent and very fair criticism of our study design that, yeah, we do have. So I think generally participants in our study tend to be wealthier. I think white individuals are overrepresented. So yeah, I think this is definitely a very fair criticism. Obviously, we are limited in what we can do to address it now that data has been collected. I think one way to address this, because the data source is so rich, we do have data on things like deprivation indices, or obviously we have genetic ancestry and self-reported ethnicity data. So we do have data that would enable people to correct for this sort of biases. But yeah, unfortunately, it is true that our cohort is perhaps not fully representative of the population as a whole.
And there was another question as well, which says, is the UK Biobank just a data provider, or does it have a role in the governance, quality, and ethics of how the data is used?
So I think definitely in terms of governance and ethics, the way we distribute data is so researchers apply to use our dataset with sort of defining the purposes and the scope of their project, which gets reviewed by our epidemiology team and our legal team if necessary. And only when the project gets approved do we release data to them. In terms of quality, we don't really tend to review the quality of publications resulting from that data set. Obviously, that's more for peer reviews. So yeah, but in terms of governance and ethics, yes, I think definitely we do consider those things when releasing data to researchers. Yeah, it's an important part of the approval process.
Thank you very much. I think we'll probably wrap up there, but big thanks go to Laura and Will for presenting today. Two really interesting talks, and I'm sure hopefully we'll kind of stem kind of further conversations afterwards in terms of using data, maximizing it. Just to advertise a couple of the events kind of coming up later in the year. So we've got 4 webinars organized this year from the CSC SIG. So the next one is in May, and that's around medical device security and cybersecurity. Then later on in the year, in July, we've got an opportunity for those, particularly in early career, to proffer papers and give an opportunity to present to your peers and to get feedback as well. So there'll be feedback given on those presentations. Later in the year, we've got one on how to talk IT. So in terms of working with our colleagues from perhaps corporate IT and the different language and kind of terminologies that they'll be using, how do we maximize how we interact with them? And then in November, slightly later this year, we've got the in-person clinical and scientific computing symposium, where it would be brilliant to have as many people along as possible to discuss data projects as well as kind of scientific computing and kind of more practical things as well. It would be really good to see you all there. There's also a number of other IPEM events coming up, which you'll find on the website, but here are a few. I won't read them all out, but I'll leave them up there for you to be able to have a quick look at in case you're interested. Finally, thanks everyone for coming today and listening. It would be great to hear from you in the future. Thank you very much.